Hi, and welcome to the first part of clinical microbiology. At the end of this course, you'll be able to review the microbial anatomy and metabolism of microorganisms. You'll be able to demonstrate a comprehensive working knowledge of each organism that we study throughout the semester, to discuss the major diseases caused by the organisms as well as the rationale for treatment, prevention, and control strategies. You'll be able to differentiate organisms by their morphological, biochemical, and serological features, and to select appropriate methods of lab isolation and identification. You'll also be able to evaluate lab data and appropriately identify the genus and species of each organism described throughout the course. So we're going to start with an introduction to clinical microbiology as I posted on the course website. This lecture is a review of what you would get in a general microbiology course. So hopefully everyone in this course has had everything that we're going to cover in this lecture at some point in their undergraduate education. So you all remember the bacterial anatomy, where you have a single cell, unlike a eukaryotic organism. Prokaryotes are single-celled organisms. They do contain a cytoplasm. Some bacteria will have a flagellum or several flagella. They do contain nucleic acids, there's DNA, there's a plasma membrane, a cell wall, and some organisms do have capsules. So again, we have cell wall, cell membrane, ribosomes, pili, flagella, nucleoid, plasmid, cytoplasm, your typical components of cells, with a couple of different things that are specific to the bacterium, such as capsules and spores and flagella and a couple of other things that you wouldn't normally see in a eukaryotic cell. The cell wall of a bacterium lies outside the cytoplasmic membrane. So the key thing about the cell wall in bacteria is it is very unique to bacteria and plants. Only bacterial cells and plant cells have a cell wall. Human cells do not have a cell wall, so this is something unique, which is actually important when we're going to talk about antibiotics later in the semester, because bacterial cells have a cell wall and human cells don't. Some of the antibiotics actually target the cell wall because it can go after the bacterium and kill the bacterium without killing our human cells. The cell wall allows the organism to maintain its shape. It protects that organism from bursting. It provides attachment sites, a platform for those appendages like pili or flagella. And the cell wall is what helps to differentiate our gram-positive bacteria from our gram-negative bacteria. The cell wall is made up of peptidoglycan, which peptidoglycan is found in the cell wall of all bacteria except for mycoplasma. And the reason is because mycoplasma is the only bacteria that doesn't have a cell wall at all. And we'll talk about that later when we cover mycoplasma. Cell walls also contain mycolic acids, teichoic acids, but only in gram-positive bacteria only, and lipopolysaccharide, or LPS, which is only contained in gram-negative bacteria. So whenever you see in microbiology a very unique feature, that's something important and likely something you should know and really store in your memory banks. So the fact that gram-positive bacteria have teichoic acids and gram-negative bacteria have lipopolysaccharide is something very important when we're going to be discussing bacteriology. So lipopolysaccharide, or LPS, it's also called endotoxin. Again, it's only created by gram-negative bacteria. You won't see it in gram-positive bacteria. The other thing about LPS is it's in the cell wall and it is only released from the cell when the cell dies. That's also important later on when we're talking about antibiotics. I'm sure all of you have heard of broad spectrum and narrow spectrum antibiotics. When a person is infected with a gram-negative bacteria, you have to be very careful with the type of antibiotic you give, because you wouldn't want to give the type of antibiotic that's going to kill the cell. You'd want to give a type of antibiotic that's going to slow down the growth, because if you gave someone an antibiotic that went after and killed bacterial cells, those cells at the same time would release lipopolysaccharide, and the release of lipopolysaccharide is actually what leads to endotoxic shock. So there are people that are very ill, have bacteremias, and they end up dying of endotoxic shock just because the release of LPS from these gram-negative bacteria. So LPS is made up of lipids and is made up of polysaccharide. Here's a picture of gram-positive bacteria on the left, gram-negative bacteria on the right, and hopefully you remember that gram-positive bacteria are purple and gram-negative bacteria have a pink reddish hue under the microscope. So here's a schematic of the gram-positive cell wall. So you can see the peptidoglycan layer at the very edge, the amino acids, and then you have the cell membrane. This is an electron micrograph of a gram-positive bacterium, and you can see the layer of peptidoglycan on the outer edge of the organism. This is a schematic of a gram-negative cell wall, and you can see that lipopolysaccharide, which is present on gram-negatives and not gram-positives. Now, peptidoglycan in a gram-positive cell is much closer to the surface. Peptidoglycan is located in a gram-negative cell, but it's much thinner and it's located deeper into the cell wall. So again, another schematic of a gram-negative bacterium where you can see the peptidoglycan layer is a little further into that cell wall. Okay, gram-positive and a gram-negative cell wall. Again, gram-positive cells have a very thick peptidoglycan layer at the outer edge of the cell wall, and that's actually what makes gram-positive cells purple. Gram-negative cells have a very thin peptidoglycan layer further in the cell wall layer, and we will talk about that when we talk about gram staining, but peptidoglycan is one of the key reasons why gram-negative cells are pink and gram-positive cells are purple. One thing that I will mention when I repeat things, usually those are things that are very important, and I'm trying to make sure I say them over and over so they get embedded into your memory. So the fact that I've mentioned gram-positive, gram-negative peptidoglycan a couple of times is definitely a hint that that's something in micro that you need to know. Here is a chart of gram-positive versus gram-negative bacteria in terms of their various properties. This is under a microscope where you can see a mixture of both gram-positive and gram-negative cells. Okay, another component of bacteria is the cytoplasmic membrane. So the cytoplasmic membrane is composed of proteins. Water can pass freely through this membrane, but protons are not able to cross the membrane. So the functions of the cytoplasmic membrane, it serves as an osmotic barrier. It's the site of DNA synthesis and also the site of electron transport as well as enzyme activity. And here's a lipid bilayer that makes up within the cellular structure of the bacterium. Okay, the nucleoid of a bacterium contains the chromosome, so that's where the genome of that organism is located. The genome of a bacterium is circular, double-stranded DNA, and it has a very similar composition to human DNA. This is an electron micrograph of flagella and pili. So pili are the small appendages that come out of bacterium that allow for adhesion. Flagella are the longer appendages that come out of bacterial cells for movement and motility. We'll talk about that in a little bit. So types of pili. There are different types of pili, these little appendages that come out of the bacterial cell. There's the common pili, and these are multiple appendages all over the cell surface. These are antigenic. They allow for adhesion, so they're usually just all over the surface of the cell. There's another type of pili that has a very different function, and this is called either the F pilus or the sex pilus. Usually there's only one. So unlike the common pili that are all over the cell surface, the F pilus, there's only one of those. And what happens with the F pilus is the F pilus will come out of one cell and attach to a neighboring cell. And the reason for that is it can give the other cell its genetic material. That process of one cell giving another cell its genetic material through the F pilus is called conjugation. This is one of the ways that bacterial cells can give one another the ability to resist antibiotics. It's giving the other cell its genome, and that genome might contain a mutation that allows the cell to resist antibiotics, or maybe gives the cell some other advantage. So again, pili, common pili, usually all over the surface of the cell, and they're very important for allowing the cells to adhere to various surfaces. Flagella are very important for motility. So if a bacterium has a flagellum or multiple flagella, it is able to move from one location to another. Flagella in a bacterium are classified based on their arrangement around the bacterial cell, and flagella are also considered H antigens, and that's based on their amino acid composition. Here's the anatomy of a flagellum. You have the basal body. At the base you have a hook, and then you have the flagellin, and that flagellum is going to kind of whip around, and in that whipping around action allows the bacterium to move, sort of like a propeller. The flagella arrangements. So you can have a bacterium that's atrichous, meaning it has no flagellum at all. Those types of bacterium are considered non-motile. That's a really important component, because later on, when we're talking about different types of organisms, their ability to be motile or not is going to allow you to differentiate between certain genus and species. So atrichous, no flagella at all, non-motile. Monotrichous, mono being one, meaning one flagellum at one end of the bacterial cell. That bacterial cell can move by that flagellum flipping around and acting like a propeller. Lophotrichous, or many flagella, are many flagella but coming out of one end of the bacterium. Peritrichous, peri meaning around, is flagella that are coming out all around the cell. Capsules are another component of some bacteria. Not all bacteria can produce a capsule. Those that can produce a capsule can have certain advantages. So capsules are very useful in allowing the bacterium to store food. Another thing that's key about capsules, So if you've all taken an immunology course, you all know that there are macrophages in our bodies, and those macrophages go around and kind of gobble up debris and gobble up bad things in our bodies, like bacteria that might get in. So a bacterium that has a capsule is going to inhibit phagocytosis. So it's going to inhibit those macrophages from coming over and gobbling it up. So that is a nice virulence factor for bacterial organisms. Capsules also can block antigenic sites on the bacterial cell wall from certain types of immunological responses. It also can assist attachment to some of the host cellular surfaces. And if you were in a laboratory, or if you took general microbiology and you worked with an organism that produces a capsule, such as Klebsiella produces a capsule, you'd see that these organisms, when they're growing on an agar plate, are really mucousy looking. They're very stringy, and you could just tell they're very what's called mucoid. So when you see a very globular mucousy colony on an agar plate, you can pretty much be sure that that organism is producing a capsule. This is an India ink stain of a bacterium that you can see the capsule around it. So what will happen is the stain, called India ink, will go all over the slide, and the bacterium, it won't be able to get through the capsule, so it'll generate a halo around the organism. And that's one of the very quick and easy ways to see if an organism is in fact producing a capsule. Another component of bacterial cells, and like capsules, not all bacteria are able to produce spores. So the ability of bacteria to produce spores is one of those key characteristics that will allow you to differentiate one organism from another. So later in the semester, we're going to talk about the spore-forming organisms. What do spores do? Spores allow cells to survive in very, very harsh conditions. So I'm sure all of you have been told, don't walk around outside barefoot because you might step on a rusty nail, or maybe you have stepped on something and had to go in for a tetanus shot. The reason is the organism that causes tetanus is an organism that produces spores. That organism can survive outside in the dirt for many, many, many years. So if you happen to get a cut and from something on the ground where there was this spore-forming bacterium, you could get infected and then get tetanus. Spores also are going to aid in the classification of the organism based on the position of the spore within the cell, the size of the spore, as well as the shape of it. So here are some photos of spore-forming organisms. You could see in the top, you'll see kind of a darker area, which is the bacterium, and then kind of a light area, that's the spore. In the left bottom, you'll see what look like tennis rackets. That's actually that bacterium that causes tetanus. It's called Clostridium tetani, and it forms a tennis racket appearance because it has a spore right at the end of the cell. Spores have a very specific life cycle to them. So you have your vegetated bacterial cell that's going to replicate, divide, do its typical bacterial things. When that bacterium gets into a situation where it has low nutrients, maybe not enough moisture, it's going to start to develop a spore coat. Once that spore coat is developed, the bacterium, the vegetative part of the cell, the parts of the cell that don't matter, are going to go away, and the genetic material of that cell is going to remain inside that spore coat. Then you have this free endospore that's able to just sit around in any type of environment for 20, 30, 50 years until it then gets infected deep into the tissues, and then that spore will dissolve away, the genetic material is there, and then you can get a vegetative, actively dividing cell again, and then end up with an infection. So we said you can classify spores based on their size, shape, and location. So you have cells that have oval spores that are on the end of the cell. You can have rectangular cells that are also on the end. When the spores, not cells, rectangular spores. When the spores are on the end of a cell, they're called terminal spores. When they're closer to an end, but not right on the end, they're subterminal. When they're smack dab in the middle of the cell, of course, they're called central spores. And then you have club-shaped spores. So you have oval, rectangular, circular, and club-shaped, and the location terminal, subterminal, and central. Bacteria can also contain plasmids. So plasmids are circular, self-replicating pieces of DNA. They're present in both gram-positive and gram-negative bacteria. Bacteria can contain a single little plasmid, or it can have many, many plasmids in the cell. So again, I said that they can self-replicate, so you can have a plasmid and then all of a sudden you can have 2 and 4 and 8 plasmids. What plasmids do is they commonly provide advantages for a bacterium. So plasmids will commonly contain antibiotic resistance genes, and if one bacteria gets a plasmid that has an antibiotic resistance gene, all of a sudden that bacteria that normally would have been killed by a particular antibiotic all of a sudden can survive. So that's a nice advantage for that bacterial cell. Since I said the plasmids are self-replicating, but they do require a host cell, so they're not going to be able to replicate out on an extracellular surface, like on a table or anything. They need to be inside a cell to replicate. So again, we said plasmids are genetic transfer between organisms. So one organism can have a plasmid that has an antibiotic resistance gene and can give that plasmid to its neighboring organisms, which of course, giving any bacteria an advantage is giving the bacteria a potential to increase its pathogenicity, or its ability to cause an infection in an individual. So here is a schematic of the overview of bacterial structures that we've just went through. Hopefully you remember from your general microbiology or even general biology course where you have your taxonomy. Microbial taxonomy, you start out with your kingdom. It goes to division, class, order, family, genus, and species. And then with some organisms there are subspecies or subgroups. In this course, we are going to function on the genus and species. Every now and again, we will talk about the family that the group of organism derives from, but usually we're going to focus on the genus and the species. Another thing to keep in mind, whenever you're writing a genus and species, whether you're writing it with a pen or on the computer, genus and species have to be italicized. The genus is always capitalized, the species is always in lowercase. If you happen to be writing a genus and species and you can't italicize, you need to underline it. So those are two important things about bacterial nomenclature. So in order for an organism to cause disease, it must be able to do several things. So first of all, it has to be able to have an encounter, so it has to come into contact with a host. It then needs to colonize the host surfaces. It would then need to enter into the host, invade the host cells, disseminate throughout the host, and then cause some sort of outcome. The outcome can be symptoms such as a fever. It could be a disease such as tetanus, or we can just clear that organism. Our immune systems can come in and get rid of that organism, and we didn't even know that we had this interaction. There are two types of bacterial transmission. One is direct transmission and the other is indirect. So in direct transmission, that is person-to-person transmission, or vertical transmission, which would be mother to the fetus. So while a mother is pregnant, you can have vertical transmission between the mother and the baby. Indirect transmission is when you get an infection from either an intermediate agent or from a reservoir. So indirect transmission can be from a vector such as a mosquito. It can be vehicle-borne or fomite, where you touch a doorknob and there happens to be some organism on the doorknob, and then you get an infection. So those are the main differences between direct and indirect transmission. The bacterial shapes, these are very important when we're classifying bacterial organisms. We have our spiral organisms, which are called spirochetes. We have our bacillus, which are called rods, because they're kind of oval rod shaped. We have cocci that are circular in shape. We have coccobacillus. A coccobacillus is actually a bacillus. It's considered a rod. It's just such a teeny tiny rod, it almost looks like a cocci. The coccobacilli are the most difficult organisms to identify under the microscope, especially to someone less trained, because they do look like cocci. We do not have anything called diplobacillus, where you have two rods that are stuck together, one on top of the other. However, rods will chain together, and that's usually called a streptobacillus will chain together, although there are other types of organisms that will chain, but streps really tend to chain together. There is an organism called streptobacillus that is a chaining rod, and then we have the chaining cocci, which are streptococcus, which we'll talk about on the next slide. So again, for the cocci, you have your singular cocci or circular shaped bacterium, diplococci, which are two cocci side by side. You have your streptococci, which are a string or chain of cocci. You have your tetrads, which are four cocci organisms stuck together. You have your sarcini, which is a small clump of cocci, and then you have your staphylococci, which is a big, huge mass of clumped cocci. Kind of looks like a cluster of grapes. So here are some micrographs of your organisms. So your diplococci, or two cocci that are side by side, and we will talk more about the organisms that have these specific characteristics. So there's a streptococcus that is a diplococcus, that is classic type of feature to differentiate the organisms. You have your chained cocci, gram-positive cocci, like your streptococci, you have your organisms that commonly cause tetrads, you have your clustered cocci like your staphylococcus organisms. You can see in these pictures of microscope slides, gram-positive organisms are usually nice dark purple. Sometimes they can be slightly over decolorized though, and they can appear a dark pink color. So to the untrained individual, sometimes those will look like gram-negative cells, and that's why it's really important to have proper gram staining techniques, because you want to make sure if something is a gram-positive or a gram-negative organism, because that's really going to let you know what types of organisms you want to be looking at. Our gram-positive rods or bacilli, you have your boxcar, big, huge, bulky rods. You have little narrow gram-positive rods. You have your coccobacilli, which are your teeny tiny rods. And then there are a few organisms that have branched shape rods. These organisms can be tricky because they actually look like fungi when you look at them under the microscope, because fungi have this branching appearance to them. Here we have gram-negative cocci and then gram-negative diplococci. So the gram-negative diplococci are classically in a kidney bean shape, and there are certain organisms like Neisseria that have classically this kidney bean diplococci morphology. So bacterial morphology under the microscope is really important because it's going to allow you to narrow down what types of organisms you should be thinking about and testing for. Gram-negative rods, we have gram-negative rods or bacilli. Again, we have that coccobacilli, the very teeny tiny bacilli. There are some rods that are or some bacilli that are curved, like our vibrios have a curved form to them. You have fusiform, which are very thin and tapered at both ends, and then our spiral organisms. And here we have a micrograph of various shaped organisms. One thing to be careful of when you're looking under the microscope is occasionally you'll see these big, huge, gram-positive looking purple circular organisms. You have to be careful because your first instinct might be to call them a gram-positive coccus, when in fact they're actually yeast cells. So if you look at this micrograph right here, you have these teeny tiny circular positive cocci organisms. That is the typical size under a microscope of a gram-positive cocci. Then you have those big, huge, circular, not really uniformed in shape, those are the yeast. In this micrograph, there's a combination of bacteria, so you can see some gram-negative bacteria. Oops, I'm trying to point to them. You could see some gram-negative bacteria in the background, chained organisms. You can see some white blood cells. So you could see some PMNs and some lymphocytes here, and you can see these big, huge, gram-positive looking things, which are your yeast cells. Yeast cells tend to bud, so you'll have a big, huge circle with a little tiny little circle coming off of it, and that's classic yeast. Another thing to be careful of is we gram stain our organisms, and one of the stains is crystal violet. Crystal violet is what is that purple color, but crystal violet does tend to crystallize over time. And so sometimes you will see these what looks like a gram-positive rod, very strangely shaped, when in fact it's crystal violet. And that's crystallized, which means you either need to strain out your crystal violet or you need to get rid of it and open up a fresh bottle. So usually in the clinical lab, everything has an expiration date. Everything is monitored daily, weekly, monthly. So your crystal violet, you'd be less likely to have this happen, although I've seen it happen many times outside of the clinical laboratory. You have to also be careful in micro for artifacts. What might look like an organism might actually be crystal violet that is precipitated, or maybe some cellular debris that's leaked out of a eukaryotic cell like a granulocyte. So just those are things that for some of you, you might be working in micro and this is just second nature to you. But for those of you that maybe have never worked in a clinical micro lab, these are things that you will need to consider once you get to your clinical rotations. So again, I said it's very important to appropriately gram stain. So you want your gram-positive organisms to be purple, your gram-negatives to be pink. If you over-decolorize a gram-positive organism, many times you'll get this mixture of purple and pink. And you don't really know, is there a mixed gram-positive and gram-negative infection going on here? Or did I just over-decolorize and I actually have a gram-positive organism that in some areas of the slide looks gram-negative? Bacteria have the capacity to exchange genetic information, and there's 3 types of exchange. One is transformation, one is transduction, and one is conjugation. And here's a little schematic of the 3. So for transformation, you can get fragments of DNA that can be floating around. So maybe a bacterial cell just died and disintegrated, and so some DNA fragments might be floating in the area, and another cell can take up these fragments and then incorporate these DNA fragments into the genome. So we can actually force this to happen in the laboratory. So in some labs I used to do this where I wanted to generate a marker and give an organism resistance to an antibiotic. I would put an antibiotic resistance gene and force it to go into the cell. So it happens naturally, or you can force this to happen in the laboratory. Transduction happens when you have a bacteriophage. So what a bacteriophage is it's a virus that infects bacteria. So this virus will pick up a piece of a genome, maybe it contains an antibiotic resistance gene. It will then infect another bacterium and give that bacterium that segment of the genome. Conjugation we already talked about. That's when that F. pilis will connect from one organism to another and transfer genetic material from one cell to another cell. The stages of microbial host interactions, we mentioned this already. You have your physical encounter, you have your host surface colonization, you have your entry, invasion, dissemination, and outcome or disease. So this is key for pathogenic bacteria, or bacteria that have the potential to cause disease. So again, in order to cause disease, a bacteria has to be able to enter. It has to overcome the host immune mechanisms, which is not a very easy thing to do. It has to be able to proliferate within the host tissues and resist all of the host defenses. Bacteria are able to do this usually through virulence factors or having virulence properties. We talked about capsules. That's considered a virulence property. It will allow the bacteria to not be phagocytized by macrophage cells very easily. Producing certain enzymes is also a virulence property. So some organisms produce beta-lactamase, which will allow them to inhibit being killed by beta-lactam antibiotics. You have adherence factors that will allow the bacterial cell to adhere to a host cell, and also some bacteria can produce toxins. And so again, toxin production is a very significant virulence property. So enzyme production, I mentioned already beta-lactamase. There's many different enzymes that bacteria can produce. So there are catalase and superoxide dismutase enzymes, and those enzymes will protect the bacterial cell from being damaged or killed by a macrophage oxidative burst. So if the cell does get gobbled up by a macrophage, the macrophage will generate oxidative products to kill that cell. If the bacterial cell is able to produce catalase or superoxide dismutase, it is going to possibly be able to survive that attack. Some bacteria produce proteases, which can destroy white blood cells and immunoglobulins. Some produce lecithinase, and those can interrupt cell membranes. That can cause necrosis. Some produce hemolysins that can destroy red blood cells. So there are many, many enzymes. I mean, we can have an entire lecture just on enzymes. Keep them in the back of your mind. Later, when we're talking about a specific organism, if it produces a key enzyme that might be used to characterize that organism or a key virulence factor, we will discuss it in more detail regarding the specific organism. I mentioned toxins being a very significant virulence factor. There are several types of toxins that bacteria can produce. So something important, a unique factor is endotoxins are a toxin produced only by gram-negative bacteria. Like lipopolysaccharide that we already talked about. That's an endotoxin. It's secreted when gram-negative bacterial cells are killed, and then it's released. So again, the release of lipopolysaccharide or endotoxin can cause shock, endotoxic shock. It can lead to DIC or disseminated intravascular coagulation. It could generate fevers. So the endotoxin is gram-negative bacteria. The toxins produced by gram-positive bacteria are exotoxins. Unlike the endotoxins, exotoxins are released when the bacterial cells are alive, not when they're dead. And exotoxins will target very specific host cells, whereas endotoxin don't have a specific host cell. They'll just attack any cell. Our immune system and body, the way our bodies are made up, is a defense mechanism against any type of invasion. So our defenses, we have primary defenses such as our skin. Our skin provides a barrier from bacteria gaining entry. We have cilia on the outside of our cells. We have mucous membranes. We produce chemicals that will inhibit bacteria from adhering and invading. So those are our primary defenses. Our bodies also have secondary defenses, such as our immune system, that phagocytosis or our macrophages going around gobbling things up, and our ability of our immune cells to produce antibodies. The ability for a human to resist an infection is affected by many different things, such as the age of the person. So we'll talk about, when we talk about the different organism, that some organisms are much more commonly the cause of infection in very, very young individuals, or maybe much older individuals, because very young individuals and elderly individuals have less, their immune systems are less in the young are less developed, and in the elderly the immune system just doesn't work as well. So age is a major factor of being at risk of an infection. Nutritional status. Malnutritioned individuals are much more susceptible to getting disease. Disease status. Individuals that have specific diseases are at risk. So a cancer patient who's undergoing chemotherapy hardly has any white blood cells, so they really don't have an effective immune system. Those individuals are at very high risk of infection. Drug and alcohol use. So alcoholism is highly associated with infection. I already mentioned chemotherapy and cancer patients, and then individuals that have foreign indwelling devices such as catheters. Anytime you have a foreign or plastic substance that is connected to the body, that could invite organisms to come in and generate a biofilm, which we will talk about later, and cause an infection. So we said that the host has defense strategies, our skin and our immune system. Well, the microbes also have defense strategies, and their defense strategies are the fact that they can multiply rapidly. E. coli has a 20-minute doubling time. Microbes can also destroy some of our host immune responses. Some of them have the ability to reside inside sites inside of our body that our immune system may not be able to get to as easily. Some organisms can produce antigenic variation, where they're constantly varying the antigenic properties on their outer surface. So our immune system, right when our immune system recognizes it and starts to produce antibodies, it'll change. Some microbes also produce virulence factors. We already talked about enzymes, capsules, spores, things like that. I mentioned how important gram staining is. So gram staining was developed by Hans Christian, and it is one of the key ways to differentiate gram-positive and gram-negative bacteria. And that's really important because you're narrowing down any test you can do to narrow down what the organism is, brings you closer to diagnosis. So for gram staining, you have your primary stain, which is crystal violet. You have Gram's iodine, which is the binding agent. You have a decolorizer, which is an alcohol that's going to remove the crystal violet from gram-negative cells. And you have a secondary stain, which is your safranin. You should look at these definitions because you should all be comfortable with the various words we're going to use throughout the semester. So just because someone has an infection does not mean they have a disease. An infection is just there is an organism inside the host. We have many, many, many bacteria that live inside of our body that never cause any problems. But we also can get organisms inside of our body and again, maybe not have a problem or cause disease. So an infection is there's an organism there. A pathogen is an organism that has a higher ability to infect a host and cause disease. Virulence is the ability of an organism to cause damage. Pus is that oozy material that can leak out of wounds, which contains white blood cells and cellular debris. And pus is the hallmark of acute inflammation in a human cell. You can read through some of these other definitions and just familiarize yourself with the different terminology. If you have a medical dictionary, anytime you come across a term that you may not be familiar with, you can also look it up, and there's plenty that are available online. For this slide, I'm just going to talk about opportunistic. Opportunistic are organisms that normally would not cause a disease, but in the right circumstance, so maybe a malnourished individual or an individual that has some other diseases that's making the immune system not work as well. Those are called opportunistic bacteria, and they might cause an opportunistic infection. So we'll talk about those later in the semester as well. We already mentioned that there are bacteria everywhere. So commensal organisms are organisms that live either on or inside the host, and neither the bacteria or us has any harm, nor do we really benefit. They're just there. Symbiosis is when an organism lives on or within a host, and both the organism and the host have an advantage or a benefit. Parasitism is when an organism lives within a host and the organism benefits, but the host is harmed. So the end of this lecture, just going to end with respect bacteria. They're the only culture that some people will ever have. If you have any questions, you can post them on the discussion forum on the discussion board in Blackboard.